Hello, so in this video, I'm going to go over how I kind of study for microbiology and just some study tips and things that I do often that I just use all the time when studying for microbiology. So this is kind of like how my setup kind of looks almost every single time I'm studying. I print out the lecture notes and I have 4 slides on one page. And so this, I make sure to do color. I'll do 4, and I'll include a lot of color-coded notes. I'll always have these pens on the side of me when I'm doing my note-taking for microbiology. I personally have certain colors that I like to use for certain things. But first of all, I wanted to explain, I like to do the printing out because I feel like instead of just seeing one slide at a time as you're going through the lecture, I feel like doing this and having 4 of your slides of the screen on one page can really help you see the bigger picture and a broader perspective. So for example, it talks about type 1 immediate hypersensitivity, and you can see all these things are related to it, and the 4 are in the frame, and to me it just helps a lot more to visualize and see the big topic. I used to take notes on an iPad, and it was one slide a page, one at a time, and I felt like just being able to look at this, for example, and then just at this at a time, I didn't get the big picture as well. So that's why I do it this way. And then I also incorporate color coding because I feel like sometimes the just the process of picking a color of what you think goes with the note already provides you're categorizing it in your mind. So yeah, I used green for a lot of the toxins mentioned. Orange for words I didn't know, red for important, yellow for vocab words on the study guide that she wants us to know. Pink was for things that stood out, like mnemonics or things, tips to remember on how to remember something. And I have a blue pen, and I would use the blue and black pen just for regular note taking. Okay, so that was mainly for the color coding and how I take the notes, and really this doesn't take too long. I mean, I just have my lecture, I'll have my video playing, and I'll basically,
the job of the lymph nodes,
I'll be having this,
filter the lymph,
have the matching slide open, and just be taking notes at the same time, just like this.
If there's bacteria or viruses.
In addition to this though, I usually have a Google Doc, and here's an example. I have my 3rd micro exam Google Doc here. And so I'll take notes on 4 slides at a time or 2 pages at a time, and then I'll pause and I'll go to the study guide and I'll look to see which terms or what of what I just watched is on the study guide. Since it's a lot more fresh in my brain, since I literally just watched it and took notes on it within the past few minutes, I then try to fill in as much of the study guide as I can. If it's something that I was kind of okay, I got that, I don't need to spend too much time typing out the definition for it, I just write on the side oh, it was on slide 5, it was on slide 3, page 4. That way later on, before I study for the exam, I can just fill it in really quick as a refresher. But if it's something that I was okay, I kind of, I could write a bit about this so that I can remember more detail in the future, I'll make questions out of my notes. So, for example, let's just say I'm watching the video and I'm taking notes, but I already finished this whole page and, I took my notes and I have them. I might even do this one, but since this one kind of covers the lymphatic system in just these 3 slides, I would pause and then I'd look on my study guide to see if there are any questions on the lymphatic system. So I would go chapter 12. And then I would look for the questions. And if there is one on what is the lymphatic system and the different functions and the organs, I would basically turn these definitions that are just stated into a question. So for example, you know, I would look at it and kind of just like analyze how I can turn it into a question. So an example of a question, of making this into a question would be like, so we have the organs of the lymphatic system, and then it says primary lymphatic organs, secondary lymphatic organs, and it states the primary ones are the red bone marrow. It gives a definition of the red bone marrow. The second primary organ is the thymus, and it gives a definition of the thymus. So what I would do is I would try to incorporate this, and the definition, so all into a question. An example of one I just did was, which of the primary organs, so we included the primary organs of the lymphatic system, produces all the blood cells and is the site of maturation for B cells. And that's the red bone marrow. So I would kind of do like questions as I go, and especially mainly only the ones that are on the study guide, because doing the notes and watching a lecture was pretty much good enough for all the other material. So for your study guide, you know, try, well, I would always do like question and answer. So I would create my own questions and then answer them. But I would try to do my best to actually reword it in a way that incorporates a lot of information, and then make these questions while the information is still fresh in my brain, because I feel like you can put you can put a lot of detail into it if you had just watched it and just learned about the material, as opposed to like trying to make these questions 2 weeks from now before the exam. Like a lot of this is it's gonna be harder to make the questions. So I do like a question and answer base, and for the things that are like you have to be able to write out as like not a multiple choice, but just like an essay question, I do take a different approach for these. But I first create a Google Doc for all of these, and I also put these in the Google Doc, but I do something separate for these. I add them to blank sheets of white paper, especially if they're a list or like a diagram or a process. I feel like this is necessary to be able to recall that information and be able to write it out. So I get blank sheets of white paper, just basic printer paper, and I will basically do like a formula sheet, or like some people call it cheat sheets, formula sheets. So this is mine for chapter 12. I normally have one for each chapter. So this one is describing the process of phagocytosis, and I basically write out the processes. I draw some pictures on the side, and I write it out how it makes sense to me. And I do this all in detail. I try to do in detail in my doc first, right after I learned it. And then sometimes I'll do this drawing right before the exam, or like, you know, while I'm studying for the exam. So like within like the past 3 days before the exam, so that I have that second pass at the information. And then when it comes to like processes though, or things that I feel like, so these are like lists, right? Some of these are processes, but in microbiology, sometimes we'll get a lot of things we'll have to explain, and sometimes it's often like a process that you have to visualize. So I do take a different approach for those. But before I get into that, I just did want to kind of go over this and explain how, like for this one, for example, describe the process of phagocytosis. Right after writing it out, I often like notice like, okay, this actually makes sense and I understand it now. Like all these pictures, like right now it's fresh in my mind. And this will be like, 2 weeks before the exam, because this is when I'm watching chapter 12 lecture, you know. Sometimes the lectures, we have like 5-6 lectures until our exam. So what I would do sometimes is like try to say this out loud without looking at this. Well, first with aid. I write this all with aid, like looking at my notes, trying to make it as detailed as possible. And then after it's written out, I try to practice saying it without aid. Obviously I'll look at it every once in a while, but I try to understand it and say it out loud. And sometimes I'll even use voice memos to record myself saying it once I like mastered like the process. or once I feel like I really got it down, I'll voice memo record myself, and I'll have that 3-minute recording later for the exam. So sometimes I do this like 2 to 3 weeks before the exam, which is when I'm watching this lecture. And then when the exam comes around, I have this 3-minute recording on the process of phagocytosis in my phone, and I'll just listen to it when I'm studying this part for the exam later on, and it all comes back, because I feel like if I hadn't done that recording or made this, sometimes a lot of that information I just forget a lot of the details, and that all those details help make it easier for me to remember. So recording it and having that recording with you so you can have it before the exam was really helpful for me. And I listened to it maybe like once or twice, and if I was able to recall it without the audio again, like listening to it and then being able to recite it, I was like, okay, I crossed that off my list and I continued to the next thing. So it's like a building up process, but it kind of like for me had to be done first right after I watched the lecture, and then again before the exam, you know. So it is a process, but I feel like it's a very helpful process, just to have all the information you need for the exam. And for the processes that were more visual, for example, like the how helper T cells are activated in chapter 13. For these, I took a more different approach in actually like drawing the picture out a bit more. To me, this was really helpful. I drew out and then I put the steps around it, like kind of like in order, and I put some definitions, just some facts on the sides that had to do like similar to this, just to keep in mind. So like, for example, something I kept needing to have in mind while learning about this was that all antigen presenting cells had MHC2 proteins. So they were those special immune proteins that had the MHC2, and all the nucleated cells had MHC1 proteins. So you found MHC1 on all the nucleated cells and MHC2 on all the antigen presenting cells. And then I keep these facts in mind, that to activate the helper T cell, if the APC displays MHC2, it activates the helper T cell. If the APC displays MHC1 and MHC2, it activates a cytotoxic T cell. So I kind of just put facts up here, and then I started the process down here. And I would write this out again with all my notes. My lecturer had just watched it, and having this out, I put a lot of detail, as much as I can like think of and put together, and I had this for the exam. But the difference with this one that I tried out for the first time actually was recording myself just like this, and I would be like, so I finished this and I wrote it out and everything, and I would be like, okay, so step 1, the microbes are engulfed by this antigen presenting cell. Then they put the antigen on the MHC2 protein receptor, and then I was like, and then you flip it, and this is now here. So you grab it, you flip it, and your helper T cell is here, and when they bind, it releases interleukin 1, and then that releases interleukin 2 when stimulated. So I would kind of do like a little video, literally how I'm doing right now is like a perfect example. And it was like 3 minutes, and it was just me explaining this process at the time when it made most sense to me, which was normally right after I watched the lecture and took my notes, made my questions, and then I made this. And I only did this like for 5 different topics. It wasn't like the entire study guide. It was just like the more detailed explained topics that we needed to know that I did this for, and it really helped. So I had this video of me explaining it, and it's in my gallery, it's in my phone. I can literally watch it anytime. And it was really helpful to have before the exam, because I watched it like 2 times. And yeah, I was able to answer all the questions on this. Very helpful. I underlined like basically like the gist of what happened in each step. And this was kind of what I did for those blue questions on the study guide that needed to be defined and needed to be, and were possible essay topics. So yeah, that's pretty much it. I have one of these blank sheets for like, sometimes I had two, so since this one took up a lot of space, I used two, but I normally just have one for each chapter. And I kind of call them cheat sheets. So like, if you could take a sheet to cheat on the exam, which one would you, like, what would you put on it is kind of like, you know, you would want like a process there. You would want the things that you're struggling with to be on here. They're also known as like formula sheets. But yeah, that's pretty much it. I hope it helps. And yeah.